Hello my dear children, Namaste and welcome to yet another session of our newly launched ICSE series for class 10. I'm Ambika, Master Teacher of Biology right here at Vedantu, and as you know, we have already started with your very first chapter, Cell Cycle and Cell Division, and on the very first day we had an amazing live session. All of you participated amazingly and gave me a lot of answers to the quiz that we had, and we saw what a cell cycle is basically. And in our second session we have seen what you were really looking forward to, mitosis and its stages, right? We have seen why mitosis is relevant to us in everyday life, also where it happens and why it is important to understand mitosis, right? So let's get started with the very next process, which is another type of cell division called meiosis. Okay, but before we start, let us look at the homework question that I gave you yesterday, which was, like I said, a very objective question. I told you already that I won't be able to select a best answer, but I was surprised to see that a lot of you came up with answers very quickly, and I was really happy to see that. Identify the following stage of mitosis, and a good majority of you answered it correctly as metaphase. Yes, this is indeed metaphase, because as you can see here, this is the equatorial line of the cell, or the middle of the cell. These are the two poles, and you see that the chromosomes are well aligned along the equator, or the middle of the cell. And if you recall, we also said that the prefix meta in metaphase stands for middle, wherein the chromosomes are arranged along the center of the cell, right? Just for a quick recap, let us also look at this. The four major stages of mitosis, right? On the first day, in our first session, we saw how interphase happens in G1S and G2 phases. Right after a cell has completed its G2 phase, what does it do? It jumps right into prophase. Pro again is before or first, right? Prophase followed by metaphase, followed by anaphase, wherein the sister chromatids are pulled apart, and finally telophase, wherein the nucleus begins to reform in order to prepare to form two daughter cells. And finally, after this, there is an additional step, which is cytokinesis, which is the last step, wherein splitting of cytoplasm happens, finally pinching off this cell into two new daughter cells. So we have a diploid parent cell and two new diploid daughter cells. That is the basic summary of mitosis, right? And why was it important? Well, we did discuss all of that. This happens in most of the cells in your body, right? Growth, repair, nothing would happen without mitosis. So let's move ahead. Now, the human body, our body, if you think of it, is made up of, any idea how many cells we have? Well, the fact is that we are made up of trillions of cells, okay? And how come we have so much? All of us started our life as one single cell, and it is as a result of repeated mitotic divisions that we have so many cells in our body today. Okay, but human cells as a whole, as far as the number of chromosomes in the cells is concerned, may be of two types. Human cells may either be diploid or haploid. You must have heard of these terms in your lower grades, but just in case you can't recall them, let's just quickly revisit these terms. Okay, diploid and haploid. Now, what's the number of chromosomes to do with? Why is it so important? Now, if you understand, if you have really understood the structure of a cell, you know that the most important part of a cell is its nucleus, right? And why is it the most important? It is mainly because it is your nucleus that contains all the genetic information, all your hereditary information in the form of DNA. And at the time of cell division, this DNA or your genetic material can be seen in the form of systematically arranged chromosomes. So a typical human cell is supposed to have 46 chromosomes or 23 pairs of chromosomes. Okay, in the case of a human being, this number would be 46, the diploid number. What about the other type, haploid number, or N? I have represented it as N, or we generally represent it as N. And the haploid number in the case of a human cell, of a haploid human cell, would be obviously the half of this, which is 23. So N, or the haploid number of chromosomes, is 23, while 2N, or the diploid chromosome number, is 46, or 23 pairs. Okay, so again, it may be a little confusing. Now, I gave you these numbers only for you to understand the meaning of diploid and haploid. Basically, just remember that haploid would have half the number of chromosomes as a diploid cell. Now, where do you find haploid and diploid cells is the next question, right? Now, in the human body, most of the cells in your body, like your skin cells that you see here, your liver cells, your neurons, your stomach cells, your kidney cells, most of the cells in your body are diploid, a good majority of them. There is only one type of cells in your body which is haploid, and that is the gametes, or the sex cells. What do I mean by that? These would be the sperms in the case of males and eggs in the case of females. So in a female human body, apart from the egg, there would be no other haploid cell. Every other cell would be diploid. And in the case of a male, in the case of a man, it would only be the sperms in his body that are haploid, and every other cell would be diploid, right? So think of it, remember the meaning of the terms diploid and haploid, okay? And remember the examples for them. Just remember that only your gametes are haploid, all the other cells in your body are diploid. Okay, and we call them somatic or body cells in general. All the cells in your body that are not your gametes are called somatic cells, or your body cells in casual language, body cells. Okay, so just keep that in mind. It's important to understand this very clearly before we go ahead, right? So, moving ahead. Now, we know that the somatic cells undergo mitotic division for various reasons, right? What reasons? We have seen that in our previous session. Basically, even your nail growth, your hair growth, physical growth of your body, and any kinds of repair of any wound or damage anywhere in your body, right? All of that happens because there is mitotic division happening in all those places, correct? What about gametes? We have said that gametes are haploid. How are the gametes formed? Mitosis was a pretty much straightforward process wherein a diploid parent cell would give rise to two diploid daughter cells. The daughter cells would be clones of the parent, by which I mean that the daughter cells would be exactly identical to each other and identical to the parent cell. This was a pretty straightforward process that happened in the stages that we have seen. But to form gametes or haploid cells, another process has to happen, which is what meiosis is all about. So let's look at the details of meiosis. So what I have written here is that the ultimate aim of meiosis is basically to make the sex cells of your body, or the gametes of your body. This is a slightly more complex step as compared to mitosis, but why is it important? Why are we so interested in meiosis? If you look at this picture that's right behind me, it's a family picture. Some of you may recognize this as Simpsons, which is a very famous cartoon. Some of you may not have seen it, but that doesn't matter here. All that I want you to do right now is observe how similar or how different the children are from or with their parents. you see that the three children do have a lot of similarities with their parents, right? Like all of them have huge bulging eyes and all of them have pretty much the same skin color and all of those things. But the main difference that you can see is perhaps in terms of the shape of the head or the hair or whatever it is supposed to be. You would see that all three of them and the parents have it appearing different from each other. How does this happen? The three children born to the same set of parents. Compare this with yourself, right? You and your brother or you and your sister are born to the same set of parents. Why is it that siblings do not look exactly identical to each other or exactly identical to the parents? Why is it that sometimes you even have a character or a trait that neither of your parents has? All of this is answered through meiosis. Now, this is another family in which we have the father, mother, and the son. The son, in fact, has a mix of the characters of the father and the mother. Like, for instance, he's got ears and eyes from dad, and he's got hair like mom's. And finally, the combination of all of this makes him unique, what he is. All of this can be explained once you have understood what meiosis is supposed to be. So let's not waste any more time. Let's get started. This is basically a gist of meiosis. Now, as far as your ICSE syllabus is concerned, you don't really have to dive in to understand every single fine detail of meiosis, but it is important to understand a little more of the detail, I will be telling you a bit more than what is in your syllabus, just so that you understand the significance and you don't forget why things happen in a certain way. Okay, so this is basically an outline of how meiosis happens. Now, one more question that you may have in mind is, why is ma'am saying meiosis? We have learnt it as meiosis. Well, I also, to be very honest, grew up pronouncing it as meiosis, but I realized a little later that the actual pronunciation is supposed to be meiosis, which does sound very similar to mitosis, but that's how it is actually pronounced. So doesn't matter, as long as you don't mess up with the spelling in your exam paper. I don't mind. Pronounce it however you are comfortable with. Okay, so let us look at this. Meiosis begins with the diploid parent cell. I have told you this already. Okay, but where does that diploid parent cell come from? You may have this question in mind. When we started with mitosis, we knew that the diploid parent cell came after interphase. Here also a cell has to undergo a preparatory phase, which is interphase. And after that, once it is ready with duplicated DNA and all of the machinery necessary for cell division, it goes on to enter meiosis. So again, it starts with the parent diploid cell. It replicates its DNA, and in the first step, it would result in 2 daughter cells. And after that, there is one more additional step wherein that further divides into a total of 4 daughter cells. Okay, this is only an outline of meiosis. I'm repeating this. There are a lot more finer details coming along. Okay, understand this for now. The parent cell undergoes one round of DNA replication followed by 2 separate cycles of nuclear division, which is exactly what I have shown you in this image here. Okay, this process results in 4 daughter cells that are haploid. Okay, remember that. Very important point. We have a diploid parent cell and we have 4 daughter cells that are haploid, right? And in humans, these haploid cells, we've discussed this earlier, are the eggs and the sperm. I hope this much of the process is clear to you. Let's now dive right into the finer details, which are the basic stages. So one more thing to remember is that because a diploid cell is getting reduced into a haploid cell, or 46 chromosomes is getting reduced into 23 chromosomes in the end result, we also call meiosis reduction division, right? If you recall, what did we call mitosis? A similar comparison, a similar term that we used for mitosis was equational division, because equal number of chromosomes in parent cells and the daughter cells. Whereas in this case, the daughter cells have half the number of chromosomes as the parent cells, which is why we nickname it or describe it as reduction division. Okay, now what are the specific stages that happen in meiosis? I have listed out the names of all the stages right here. For the ease of understanding the stages of meiosis, the stages have been divided into meiosis 1 and meiosis 2. Okay, that's how everyone studies it. Meiosis 1 and meiosis 2, each individually has its own prophase, metaphase, anaphase, telophase and cytokinesis, which is why we write it this way: prophase 1, metaphase 1, anaphase 1, telophase 1, all of which are part of meiosis 1. This is followed by cytokinesis 1, following which the two daughter cells resulting from this individually enter into prophase 2, followed by metaphase 2, anaphase 2, telophase 2, and eventually cytokinesis 2, which would give you the ultimate end result, which is 4 daughter haploid cells. Now let us look at what happens in each of these stages, right? The very first step is prophase 1 of meiosis 1. Here what happens is, similar to mitosis, the chromosomes or the chromatin condenses and thickens into the X-shaped structures that we are familiar with, right? And chromosomes line up with their homologous pairs. I've explained what homologous chromosomes are in my previous sessions. Just in case you can't recall that, please do make it a point, go back to those sessions videos and do understand what homologous chromosomes are. Okay, so chromosomes line up with their respective homologous pairs, and there is a very special process that happens in prophase 1, which is called crossing over. Okay, this process occurs between non-sister chromatids of homologous chromosomes. Now this is what a cell in prophase 1 would look like. You can see the chromosomes are condensed and thickened, and the nuclear membrane has begun to degenerate slightly. It's becoming lighter. One more question that I have for you. Why do you think the nuclear membrane disappears during cell division? Well, some of you must be knowing the answer. Those of you who do not know the answer, here it is. It's for the simple reason that the chromosomes have to move apart to the poles of the cells as cell division progresses. And because this has to happen, the nuclear membrane should not come in the way of movement of chromosomes. So as soon as prophase begins, the nuclear membrane begins to degenerate. By the time the cell enters the next stage, the nucleus would have completely disappeared and the chromosomes would be released, and then movement can freely happen. Okay, now let us understand a bit more about crossing over. Now I have told you homologous chromosomes pair up together during prophase 1. Let us look at this particular image. Okay, say for example in a typical human cell, okay, there are 23 pairs of cells. Now why do we call them 23 pairs and not 46 chromosomes? 46 is not wrong, but 23 pairs is more appropriate because most of them are paired. They are paired as their paired with their homologous corresponding chromosome. And in one pair of chromosome, in your first homologous pair, you have got one member from your mom and the second member from your dad. Say for instance, this is your chromosome number 1. One homologous pair. One member, the green member, say you have got it from your mom. The red member, you have got it from your dad. Okay, they are homologous chromosomes because they carry alleles or genes for the very same characteristic. Well, it may seem a little confusing now, but as we go and study a little more about chromosomes and genes, I'm sure all these concepts will make much more sense to you. For the time being, to understand meiosis, just keep in mind that homologous chromosomes are similar chromosomes that contain genes or carry genes for the same characteristic feature. Okay, so now if you look at this pair of homologous chromosomes, you would see that sister chromatids, the green sister chromatids, are held together by this centromere, and the red sister chromatids are held together by this centromere. Okay, so in this homologous pair, the green ones are sister chromatids, the red ones are sister chromatids. What happens in the process called crossing over is that genetic information gets exchanged between non-sister chromatids, which means one green chromatid and one red chromatid. As you can see here, crossing over or exchange of genetic information happens. And once this exchange has happened, what you get as a result would be this. Just imagine it like this. One piece of this is removed and fit into this. Okay? Picturize it like that. It will be much easier for you to understand. It's basically exchange of genetic information or exchange of DNA that happens between non-sister chromatids of a pair of homologous chromosomes. And this is what you call recombinant chromosomes. Recombinant chromosomes, because some kind of recombination of genetic material has happened and you have a completely new combination, which is this. So the process is called recombination. The end result of crossing over is called recombinant chromosomes. This process of exchange of genetic material between non-sister chromatids of a pair of homologous chromosomes. Understand, children, pay attention. This is very, very important to understand. This process is called crossing over. Okay, so let us now look at what happens next. Crossing over, as you can see, I have put it here, results in recombinant chromosomes. So as meiosis progresses, it will be the recombinant chromosomes or the chromosomes with the newer characteristics that will progress and proceed to the next stage, right? So after this, we have the next stage, which is metaphase 1. And what happens here, just like in prophase, here also meta refers to middle. And as you can see in this picture, these are the two poles of the cell. This is the middle or the equatorial line of the cell. And these are the recombinant chromosomes, which are still in pairs, okay, that are lined up along either sides of the equator. Okay, these are the recombinant chromosomes which arrange themselves along the equator of the cell. This is what happens in metaphase 1. Coming to anaphase 1, what happens is that homologous chromosomes or the recombinant chromosomes as they are now get separated and pulled away towards the opposite poles of the cell. Again, recall that ana means opposite. Members of the homologous pair get separated and move towards opposite poles of the cell. Okay? And what is it that pulls them apart? It is the spindle fibers. These thread-like structures that you see are the spindle fibers which have been arranged from the centrioles. These structures are the centrioles. Okay, so we have reached anaphase 1 successfully, prophase 1, metaphase 1 and anaphase 1. What happens next? Obviously, it is telophase 1, wherein the nucleus begins to reform in order to prepare itself to form two new daughter cells with 23 chromosomes each. Okay, but it's not divided completely yet. You see that some division is beginning to happen, the furrow is beginning to form, but they have already formed individual nuclei. All right, and this is followed by the final separation, which is cytokinesis 1, wherein the splitting of cytoplasm occurs here, and you have the end result at the end of cytokinesis 1. This is what you would have. You would have 2 new cells with 23 chromosomes as opposed to 46 chromosomes that the parents had. Okay, so remember where we started. Reduction of the number of chromosomes has already happened here. Now the thing is, we are at a stage where we have 23 chromosomes in these cells. We have 2 cells with 23 chromosomes each. And now you see that the cytoplasm splits up into 2 new cells with 23 chromosomes each. But if you think of the number of chromatids, it would be 46. And that is because the chromosomes are still present in this manner. So this is 1 chromosome, this is another chromosome, but although I have drawn only 2 chromosomes here, you can see that there are 4 chromatids. And the next step is to separate out the sister chromatids also, just like what happened in mitosis. So now let us begin to understand that. Now, if you have understood meiosis 1, children, meiosis 2 will be much simpler because it is very similar to mitosis. Okay, meiosis 2 is very similar to mitosis. So for now, let us look at the progress that we have made so far. These are the different stages that happen in meiosis, and meiosis 1, we have gone through so many stages, and in all these stages, what was the most eventful stage or the most interesting stage that we learned today? I must say it is prophase 1 because a special process happened there called crossing over, which resulted in recombinant chromosomes, right? Now that we have completed cytokinesis 1, we have 2 daughter cells which are individually ready to enter prophase 2. Now, how does that happen? Now, in every slide, in every image that we will have 2 cells because we are talking about both of them simultaneously, right? The 2 daughter cells we have already got. The chromosomes appear condensed and thickened, and spindle fibers again newly begin to form, just like prophase of mitosis, right? The next stage would be metaphase 2, in which in both of these cells, the chromosomes where the sister chromatids are still attached to each other, align themselves along the equatorial plate or the middle of the cell, right? And in the next stage, just like mitosis, here it's called anaphase 2 because it's part of meiosis 2. The spindle fibers begin to pull away and separate out the sister chromatids. Okay, so at the centromere, the separation happens and sister chromatids are pulled apart and move towards opposite poles of the cells, right? This happens in this cell as well as in this cell. And finally, in telophase 2, you see that they have completely moved towards opposite poles and their nuclei have begun to form, right? So finally, when cytokinesis 2 happens, this is the end result. Wow, that is a lot, right? We have 4 new daughter cells here, and each of them has 23 chromosomes and 23 chromatids, which was the ultimate aim. So meiosis had to happen as meiosis 1 in order to separate homologous chromosomes, and meiosis 2 had to happen so that the sister chromatids could be separated out, and ultimately we have an end result of 4 haploid daughter cells. And what do these haploid cells become in our body? We have seen this at the start of this session. They become the egg cells in females and they develop into the sperm cells in the case of human males, right? I hope this is very clear to you. Remember, we have 23 chromosomes and 23 chromatids in each of these daughter cells. Very, very, very important point to keep in mind. So yes, wow, that is a lot of progress. Starting with prophase 1, metaphase 1, anaphase 1, and so on until we completed cytokinesis 2. You have learned this much today, and that was indeed hard work, I must say, and I hope you have this very clear in your mind now. Now, one common confusion that children have, okay, I would like to tell you at this point. For me also, as a student, there was a lot of confusion while studying meiosis, because homologous chromosomes, sister chromatids, what is the difference between all of these? Well, I have only one answer to you at this point. The simplest way I can tell you this in one sentence is, homologous chromosomes are separated in meiosis 1. Sister chromatids get separated in meiosis 2. But how this happens, please go to the start of this video and watch it again and again until it is completely clear in your mind how the process happens. Do not get mixed up between sister chromatids and homologous chromosomes. Okay, very important, children. Do keep this in mind. So this is just a summary of how meiosis happens, all the stages. In the previous slide, I just had all the names. Here I have all the stages for you put in. You can just take a few seconds, a couple of minutes if you want, and have a look at this. Interphase, and then after interphase, it enters into prophase 1. It is still a diploid parent cell. And then metaphase 1, anaphase 1, telophase 1, cytokinesis 1, wherein reduction has already happened, and these two cells enter into meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2, and finally we have 4 haploid daughter cells. Okay, so now what you need to remember is the definition of meiosis. I think now we are in a position to define meiosis as a process. It is a type of cell division that results in 4 daughter cells, each with half the number of chromosomes of the parent cell, as in the production of gametes. Okay, this is very important just in case you are asked to define meiosis and what its significance is. You will have to write this first, and the significance, yes, we will be discussing that also. Now, coming back to this family picture. Now, can you think of any reason why the children look different, a little different from each other and a little different from their parents? And this boy here, why is it that he has a combination of characters of his dad and his mom? Well, the magic lies in the step, the special step that we discussed in prophase 1, which was crossing over or recombination, wherein exchange of genetic material happened, and finally you had a recombinant set of chromosomes which had a mix of genes from the father's genes and the mother's genes. Recombination or crossing over is the answer to these questions, right? It's also the answer to the question why you and your brother or your sister look different from each other and look different from your parents. Well, just in case you're wondering why identical twins look exactly like carbon copies of each other, that is a completely different question which we will be answering as we study the chapter reproduction, and we will also discuss a little bit of the details in the chapter genetics. So stay tuned, do not miss any of those sessions. I will also be relating to the topics that I have discussed in today's session. So do have all of these analogies and comparisons in mind also. Okay, so here again I have listed out the significance of meiosis just so that it will be easier for you during your exam to recall. The most important significance is that the chromosome number is halved so that the normal chromosome number of a species can be maintained with every generation. What do I mean by this? Just in case you were wondering, why should a sperm cell and an egg cell be haploid? Why can't we less complicate things and have diploid sperms and diploid eggs? Why do you think so? Now, the answer lies in this. Now, because a sperm cell is haploid and an egg cell is haploid, during fertilization, they come together to form a diploid zygote. And only if the zygote is diploid would it be just like any other normal member of its own species. Only this would be a normal human baby. Only this can develop into a normal human baby. Okay, this is how all of us started our life, as a diploid zygote cell. Okay, so imagine, just in case the sperm had been diploid and the egg cell had been diploid, the zygote would have had 4 sets of chromosomes, and with every generation this would have kept on multiplying, and eventually we wouldn't look anything at all like our parents, our grandparents, or our great-grandparents. With every generation, I think there would be like quick evolution into new species or newer creatures coming up, right? So it is important that the typical chromosome number of a species is maintained, which is why gametes are always haploid. And in order to get haploid gametes, our cells have to undergo through the process called meiosis. This is all that I have explained in the first step here. And in the second step, I have written that meiosis results in genetic recombination as a result of crossing over. This leads to variation. Now, if genetic recombination did not occur, if crossing over did not happen, all of us human beings would look exactly like carbon copies of each other. Right? We all look different from each other. Each of us is special and unique only and only because of the process called crossing over that happens in meiosis. Okay, so maintenance of the number of chromosomes and having a lot of variation or recombinance are the 2 major significant aspects of meiosis. Okay, so this, I just put it there because I found it a fun way of comparing mitosis and meiosis. Thanks to Amoeba Sisters for coming up with such a creative GIF. This is just saying attack of the clones, mitosis, right? Because the chromosome number is exactly the same in the parent cell and the daughter cells. Meiosis, it says, dare to be different. See, they are all different from each other and they are different from the parent cell also. 46 and here it's 23 each. Right? So that is how different mitosis and meiosis are. Now we have spoken about a lot of things. I know there's too much in your head, but I would like you to think over it and come up with a good explanation and a good answer to this homework question, which is, what are the major differences between mitosis and meiosis? I don't want a bookish answer. I want you to understand mitosis and meiosis completely and come up with a very good explanation. I'm not going to be giving you marks for each point, so the number of points doesn't matter, but please understand both very clearly and try listing out and explaining what the major differences are between these 2 types of cell divisions. Okay? Do not forget to download Vedantu's learning app and do join our Telegram group right away today if you have not done it yet. And very importantly, if you found this video useful, do remember to hit the like button. Do remember to share it with your friends who you think it may help. And in case you have not subscribed to our channel Vedantu 9th and 10th, do hit the subscribe button right now, because we have started with our hardcore chapters. The chapter Cell Cycle and Cell Division is almost done. We will be taking up more topics in the days to come, so do not miss any of our sessions. So stay tuned. I will see you again, but in the meantime, in case you have any doubts, any questions, because I agree that today's topic was a little complex. It may seem a little boring also for some of you. Unfortunately, it's a very, very important concept to understand cell division, cell cycle as a whole. In case you have any questions based on what I have taught you in the previous 3 sessions or just from today's session, do remember to put in your questions in the comment section below or drop in an email at ambika.gopalakrishnan@vedantu.com. I will see you again in another interesting session with a lot more interesting information. Until then, happy studying and bye-bye.